Well, good morning, good afternoon, and good evening, everyone. On behalf of Genelia and our 7 sister institutes, I'd like to welcome you to Life Science Across the Globe. My name is Ron Vale, and I'm the Executive Director of Genelia Research Campus, which is part of Howard Hughes Medical Institute, and we're also the organizer of this series. So today's event is a very exciting one. It's on connectomics, and it's being hosted by our sister institute, the MRC's Laboratory of Molecular Biology in Cambridge, England. And today we're going to have 3 short talks followed by a moderated discussion and audience Q&A. And please go ahead and write your questions in the Q&A box at any time. And we'll also end with a Meet the Panelists session. And this is a great opportunity for students and postdocs and other trainees to stay on the call and ask questions and hear career advice from our panelists. I'd also like to encourage you to please complete the brief survey that we'll link in our chat box, and your feedback is always appreciated. And now I'll turn it over to Greg Jefferis, who is group leader at the LMB. And Greg is also the moderator of today's session. So Greg.
Thank you very much indeed, Ron, for the introduction and overview. So as he said, I'm speaking to you from the MRC-Laboratory of Molecular Biology in Cambridge, today's host institution. So our theme is connectomes, what they are and what impact we can expect them to have on our understanding of the brain and the development of AI. And we've got 3 great speakers to cover those topics from different angles. The theme's appropriate for the LMB, not only because it's currently a hub for global collaborations in connectomics, especially in Drosophila, but it's also the birthplace of connectomics. And that's down to the vision of Sidney Brenner when he was at the LMB. He decided that it would be useful to reconstruct the shapes and the connections of every neuron in the brain, or at least the nervous system, let's say, of the nematode worm C. elegans. So that was done at the time by an awful lot of manual labor, tracing neurons by hand from electron micrographs. Much of that work was done by Eileen Southgate, pictured here. When they finally published The Mind of the Worm after about 15 years of work in 1986, she took early retirement. She was done with science. And to be honest, connectomics kind of took a bit of a pause for the better part of 20 years after that. And it was actually thanks to our first speaker, Moritz Helmstaedter, and his mentor, Winfried Denk. They were really instrumental in the rebirth of connectomics as a modern science. So Moritz has had an incredible run of science contributions and career advancement since then. And he's now director for some years now at the Max Planck Institute for Brain Research in Frankfurt. So over to you, please, Moritz. We're looking forward to your talk.
Thank you very much for this very kind introduction, and it's a great pleasure to be here and talk about connectomics from our point of view, which is focused very much on the cerebral cortex and which uses techniques involving electron microscopes. But before describing this strategy of approaching brain's circuits and their use, I want to briefly set up why I think it's so important to do connectomics at the synaptic level and analyze circuits that store information about the world in relevant ways. So many objects and phenomena in our world have been around for a long time, millions of years. So certainly on time scales that allow, in principle, these kinds of phenomena to have consequences on our genomes, and by genomic information be implemented into our neuronal circuits in terms of what can be expected in the world. Obviously, gravity, movement of objects, smell are phenomena that have been around on these timescales of millions of years. But then there are, of course, also phenomena that we all experience that are certainly not as old, and that certainly do need to come into our brains, so to say, during our lifetimes, and more generally during individual lifetimes. And it is both of these kinds of information about our world that we think end up in our connectomes, one via the genomic pathway and one by direct learning, if you want. And if you want to make this very simplistic, this differentiation, you could argue there is if you want, there are evolutionary traces in our connectome. These could be rules of wiring, etc. And then there are individual traces that need to be added, especially for more complex organisms. And then all of this information now shown on the left side enters, after having entered our connectomes, is able to be combined by our connectomes, that is our neural circuits, with current sensory data from the world, visual, perceptive, sensory, auditory, to then take conclusions, identify objects, make predictions about the world, identify interesting phenomena that are happening right now. And it is this combination of knowledge or prior information with current sensory data, in my view, that is really particular about the performance of the nervous system. And when we think about today's AI, then we know quite well how to train it heavily and then make use of it. But the efficient encoding of models about the world, both by circuit structuring with already the expectation of certain phenomena, and by, if you want, memory encoding of more recent or more transient phenomena. And then the combination of that with sensory data is something that I think nervous circuits do extremely well. And this is what I would set up as the strategy for potential better AI via connectomics from our point of view. And it is clear already from that simple argument that we need to look at the circuits at synaptic level, because as far as we know, evolutionary mechanisms of circuit structuring could be implemented by cell types. But data, memories, knowledge about what happened in my own life is, as far as we know today, most certainly implemented by the change of single or many single synapses. So we need to look at circuits at that level in order to determine these contributions to neural processing. A final introductory comment is about the evolutionary aspect of this, because what I just described as genomically encoded circuit versus individually shaped circuits is something that dramatically changes over the course of the evolution. Not so much the representation space of the genome, but certainly the representation space of the connectome. So in other words, the amount of connectomes that could be in principle assumed by a given brain rises dramatically. So the y-axis is double log 10 scale, rises dramatically across evolutionary scales. And interestingly, and with this I will conclude my introductory remarks, this is only true if you look at each nerve cell separately. If instead you look at what we would call the cell type connectome, that is the connectivity between identified neuronal types, then this encoding space is not necessarily so dramatically increasing over species and over evolution. So you could argue in this very simplistic view that I've just presented that the genetically determined part of the connectome could be in principle covered by cell type connectome description. But if you want to also add information about what individually has been learned and understand how that individual component of learning contributes to sensory and other processing, then you will have to look at these other parts of the circuit which are beyond the cell type connectome. And these proportions dramatically increase, especially then in mammals and higher mammals. So with these setups, I would like to discuss now our strategy for EM-based, electron microscopy-based connectomics that has evolved over the last about 20 years. And that is chiefly based on the fact that we need to reconstruct morphologies, cable shape, cable continuity, branching cables to reconstruct connectomes from morphological data. This, of course, is one approach, and we'll hear about exciting alternative approaches later today. But as long as we want to use the cable-based synaptic level description of the circuit, then we need to actually follow these cables, image and follow these cables in 3D. For this, we take essentially biopsies of brain tissue, embed the material in plastic, make sure there are enough heavy metals around, so to say, in the structures we want to image in the electron microscope. And then one of the key techniques so far, that was the first one in the modern age of connectomics, if you want, introduced by Winfried Denk more than or about 20 years ago, is shown here as a representative of three-dimensional electron microscopy, as we would call it today, with the ability to image this specimen here from the surface of the tissue block and then have some way of getting rid of the just imaged tissue, in this case by a diamond knife that abrades the top about 25 nanometers, and then another image is taken. This, of course, 24/7 over many months to get the data set sizes we need. And this we first applied to the retina of mouse, and in a way that is the first mammalian connectome that we obtained at the time, with about 1,000 nerve cells, shown here, photoreceptors, bipolar, amacrine, and ganglion cells that provide the first sensory processing of, in this case, visual information in the mammalian brain, where, as we all know, the photoreceptor inputs are converted into more complex, if you want, filtered versions of the pixelated world, carried by the ganglion cells via the optic nerve to the rest of the brain. And the generation of these filtered visual responses happens in this inner plexiform layer of the retina. And by reconstructing these about 1,000 nerve cells and all of their contacts, we were able to provide this connectomic description of such a first sensory processing layer in the mammalian brain. This at the time was already done by a combination of human and machine analysis, and at the time already a first starting point of AI, heavy usage of AI for the processing of such kind of data. And that was then compiled into what we today call connectomes. I would ask someone to tell me, in case you are not seeing this movie. So the generation of
Cambridge.
Okay, connectome at the time was what we had planned for, to analyze these simple visual circuits. Now, relating back to what I said in the beginning, I think this is an example of genomically encoded circuitry. So by all we know, the detection of, for instance, direction of movement, direction selectivity circuit, which was one of the first circuits we looked at the time, but also all the other about 30 filtered responses of the visual input in the mammalian retina are very, very likely mostly genetically encoded circuitry. Cell types made to link together in a way that we can understand in terms of visual processing, filter generation. And I think if we think about it in this evolutionary versus individual learning, this is most likely something that has been evolved over evolutionary timescales. Now, in order to come to individually learned circuitry, we focus currently on the mouse cortex, primary somatosensory cortex, here in this case the whisker cortex, very well known also in the term of barrel cortex, where especially in this cortex the notion of the cortical column is very visible, because the thalamic input comes to layer 4 of cortex and there creates clustered representations of sensory input. And again here we can now ask a bit more in more detail how both the processing of sensory information is encoded or is carried out by our connectomic circuits, so to say, or circuits, and how sensory experience may shape these circuits at the same time. And this we exemplified by a work that was published about 2 years ago, where we took a very small piece of cortex, densely reconstructed it with all the nerve cells in there based on EM-based data. Not just the cells, which are only a few % of the path length, but especially the axons, which are meters of path length, and then constructed this cortical connectome, sized a few thousand times a few thousand, and were then using this kind of cortical data to ask about possible traces of individual learning. And this now may seem a little bit unintuitive, that you can take connectomic data, a circuit, a snapshot, a temporal snapshot of circuit structure, and still ask questions about what part of the circuit is likely, if you want, genetically encoded in terms of cell type specificity, versus what part of that circuit is at least possibly the result of individual learning. And as we know, individual learning has been associated with a phenomenon called Hebbian, or learning, and molecularly identified with the term long-term potentiation and depression in some way. So if we focus on this notion of long-term potentiation, then it had been noticed earlier on, especially by Kristen Harris and colleagues, that pairs of synapses are very interesting. Why? Hebbian learning assumes that learning or more precisely synaptic weight change, is associated with the electrical activity of pre- and postsynaptic neurons. In particular, the proposal that when the presynaptic neuron contributes to the postsynaptic neuron's activity, then these synapses between them should be strengthened. Now, the particular configuration of pairs of synapses are extremely relevant because in these, we can even in a snapshot of a connectome study whether synapses are consistent with this kind of Hebbian learning model. Why is that so? Well, the simple prediction is that electrical activity in the presynaptic axon, of course, would be the same between the two synapses of a pair of synapses. And then together with the timing predictions, you would argue that synapses grow together if LTP conditions are fulfilled or shrink together if LTD conditions are fulfilled. And LTP with a rise in synaptic weight is predicted to then make these synapses not just larger, but also more similar. And this is something we can analyze because we can now look at pairs of synapses and ask whether they are overly similar when they are large or overly similar when they are small. So in this simple depiction here of synaptic strength, y-axis is strength and x-axis is similarity between pairs of synapses and their weights, we can find or identify regions in which LTP synapses will reside and LTD synapses will reside. And by measuring thousands of these configurations, we were able to compare this to the random case and then identified regions of this plot here. In other words, pairs of synapses here shown in green that are likely the result of LTP or at least consistent with LTP and those that are not. And that's very important. We can measure for the first time, we think, the fraction of the circuits that can possibly be learned by these mechanisms of individual learning and those that are not compatible, at least not with a saturated and fully performed LTP process. And that is, I think, quite critical. Obviously, it's a very first step. It's far away from reading the content of this kind of shaping, but being able to at least determine which part of the circuit, as shown here in purple, is even compatible with individual learning is, I think, a step in that direction. This is briefly visualized here again, the density, obviously, of the data, the dense packing of neurites that we find in all of these neuropils, and then the identification of the synapses that can likely be learned or not. In the final very few minutes, I want to focus briefly on another aspect that I find relevant for what in particular we can learn from nervous circuits, neural circuits in brains about potential improvements in AI. And I will really have to summarize this very briefly with a brief interruption earlier. But the key point here is that inhibition is something that so far AI has not embraced, at least not to any substantial degree, and is something that we need to understand, I think, and might be a source of very relevant computational properties that we could at some point potentially use. In especially mammalian brains, inhibition is dedicated to certain types of neurons. It's not the same neuron that can be excitatory and inhibitory, which of course in other species is well possible, which leads obviously in the first instance to a decrease of overall neuronal activity because you can encode with a sign times activity. But there's another aspect of this inhibitory wiring that we found a few years ago in which we found that the circuits are shaped very precisely in a triadic fashion where inhibition is always activated first. So by placement of synapses along axons, and really I have to summarize this very briefly, this is published work, but I want to focus on this triadic circuit structure here, where excitatory neurons, whenever they target the next excitatory neuron population, first make contact with inhibitory neurons that then project very efficiently to that same excitatory target. In other words, a gating circuit that we could at least show in principle could be a shut gate where activity cannot be propagated because inhibition is so fast and so strong by a synaptic, if you want, a bias towards inhibition, as wider axons, synapses made earlier, more synapses made, that you could have a shut gate. And such a triadic circuit with a shut gate configuration by inhibition could in principle compensate for Day's law, where inhibition needs an action potential to become active, and could be a configuration in which certain activity processes are encoded by these kind of triadic configurations as shown here, and then could be locked, could be unlocked by either disinhibition or additional contextual or sensory input that could then lead these shut gates to open. Now, this may seem very simplistic, but it's actually a potentially very powerful way of encoding hypotheses, right? That you could essentially lay down all that could happen from the current state of the world, but you don't use neuronal activity to actually run through all these possibilities. They are there in, if you want, silent anatomy, and they are shut by inhibition. But when you disinhibit or when you add more excitation, that is context potentially, then you start activating those hypothesis circuits that make sense in the current world. And these are ideas of how to analyze data that comes from connectomic analysis of mouse cortex in particular, even at scale. So we are now using larger and faster microscopes to get to what you would consider an entire cortical column connectome, and this is just the outlook. So we are now able to reconstruct circuits at the level of a cortical column, about 10,000 neurons. And this is, of course, the place where we can now really ask about individual learning, processing circuits, so to say, that are there to process this information efficiently. And as mentioned earlier, it's not enough to do this in mouse. We need to do this in human, as we are also pursuing right now, because I think this evolutionary scale is incredibly important to understand what happens the more the individual is able to adapt to the world compared to species where it's primarily about surviving in a world that has been around for millions of years, exactly like that already. And for this, mouse cortical column is already quite a step forward, but human cortical column is ahead of us and even larger circuits. With that, I hope that we will be able to answer questions about how knowledge combines with sensory information. I outlined very briefly, and unfortunately a little bit interrupted by my technology, the strategy we are pursuing using electron microscopy, and hopefully we'll learn something about how to compute that may be better than von Neumann architecture and current AI. With that, here's some summary statements. And I thank you very much for your patience and your interest in this work.
Thank you very much indeed, Moritz, for a beautiful and exciting talk. If you want to stop sharing now, I guess we'll be moving on to Grace's talk. So I'm excited to introduce Grace Lindsay, who is a computational neuroscientist, and she has had a very rapid rise in her own field. I think she's already very well known for her work there. She's currently a Sainsbury Wellcome and Gatsby Fellow based in the Computational Neuroscience Unit at UCL. She's also a very effective and impressive science communicator. And so I'm looking forward to that aspect of her talk as well. So I guess, Grace, if you're ready, we're ready for you.
Okay, so coming from the side of being a computational neuroscientist and building models and all of that, I'm going to try to tackle the question of how the connectome data that Moritz talked about collecting, how can that actually impact the development of AI? Because that is kind of the second part of the question that that we're raising here today, is how will this sustain the revolution in AI? So that implies that we're at a current place in AI, and we want to know how connectomic data can help keep that going. So the way that I see the way that neuroscience can influence AI is there's kind of a spectrum of where the influence can take place. And most broadly, we can aim to build AI that matches biological behaviors. Now, in some way, that's almost the definition of AI. It's artificial intelligence, and it's implied that the intelligence you're referring to is biological, and you're trying to form an artificial version. But beyond kind of the broad strokes, we want computers that are smart. You can really dig deep into human or animal behavior and come up with very targeted goals of what kind of behavior you want your AI to have. And you can hope that by matching the details, including the errors of biological behavior that you'll be able to design a system that, you know, is robust and acts like biological intelligence. So that's focusing just on kind of the outputs of the biology, the behavior that the organisms produce. And you don't really have to look at the brain at all to do that level of neuro to AI influence. If you go one step further, you can try to aim to replicate the algorithms that the brain implements. So kind of thinking of this as like the software level of the brain. What is it that we're actually doing that leads, you know, kind of under the hood that leads to these intelligent behaviors? Can we try to reverse engineer those algorithms and put that into an AI? And then in the most detailed level, you can actually try to just replicate the architecture of the brain. So look at the neurons and how they're connected and try to make artificial brains in that kind of direct mapping. And these kind of relate also to Mars levels, which is talked about in computational neuroscience, with matching behavior being like, you know, getting the same computational level, getting the same goal for the system. Then the algorithmic level in Mars levels would obviously be matching the brain's algorithms. And then the implementation level is the actual you know, physical architecture that the brain uses to implement those algorithms. So we can try to kind of make AI match up with the brain at any of these levels. I would say that there is a difference in how hard it is to identify the goals when you're using these different levels that you want to match at. So as I said, for the behavior side, you don't have to look at a brain at all, and that makes things a lot easier. It's not that hard. I mean, it's not that easy, but it's not that hard to observe the behavior of humans or animals in the lab or the real world and try to kind of distill that down into a goal for your AI to match. And sometimes that's as simple as having humans label images and then try to get an AI to produce the same labels for those images. On the other far end is the capturing the architecture, which means essentially getting the connectome of the brain that you're interested in replicating. And I'm not saying that's easy at all. You know, I have it here as definitely more difficult than matching the biological behavior. And Moritz showed, you know, all the steps that go into actually getting that connectome data, and it is difficult. But I would still say that that end of it is actually easier than identifying the algorithms that the brain uses, because the algorithms that the brain uses can't be read out in an EM scan. They're kind of this more abstract thing that we pull out of a combination of a lot of different data. And so in some ways, that's the hardest level to try to replicate. Now, I think connectomic data can have an influence for both of these two parts of the spectrum on the right here, the replicating brain algorithms and capturing architecture. So that's what I'm going to talk about, is the influence that connectomic data can have in those two ways of making neuroscience and AI meet up. So I'm going to start with talking about how we can use connectomic data to replicate the architecture of the brain. So to start talking about that, I kind of have to talk about where we are in the kind of AI revolution and what's used for AI these days. And that is primarily artificial neural networks. And very simply, artificial neural networks just try to replicate the very basic computations that neurons do. So you can see here, this is an example of an artificial neuron. It gets 3 inputs from other artificial neurons. Each of those artificial neurons has an activity value, which you could think of as a firing rate. And the activity of each of those input neurons gets multiplied by a weight, which is here in black, and then you sum all that up, and that gives you input to this artificial neuron. That input goes through some sort of nonlinear activation function, and then that produces an output. And the most common one that people use today is just the rectified linear unit. So that means that any input that is below 0 gets set to 0. So in this case, the input was -4. So the output of this neuron is 0, and anything above 0 is just set to the value that it is. So that's the basic building block of artificial neural networks. They were inspired by real neurons in the 40s. And so then when we talk about deep neural networks, the idea is just that you take a bunch of these artificial neurons and you stack them really deep. And from doing that and setting the weights just right, you can have a situation where you put in an input like an image, you put in the pixel values of an image, and you propagate through that through all the layers of the artificial network. And the last layer could give you an output like a label of what's in that image. That's the kind of transformation that these deep artificial neural networks can do. And the way that we set those weights, because those weights are really what define, what the network does, is through an algorithm called backpropagation, which just involves having kind of a target for that last layer of what the activity should look like when it gets a certain input. You can find the difference between what it should look like and what it does look like when you first initialize the network randomly. And then you just kind of propagate that information back through the network and update all of the weights, all of these connections between the neurons to make the network better at producing the correct output. So that's the basic structure of artificial neural networks. You can obviously see that there's a pretty decent place to input connectomic data here because, as I said, the whole function of the artificial neural network is defined by the weights between the neurons and the connections between the neurons. So it seems like maybe we have some straightforward way to put in connectomic influence. And so that's what I want to talk about. So I think you can kind of think of it as two different ways that you can take in connectomic data and put it into an artificial neural network. You can try something like an exact replica if you have the full weights of a set of neurons and you just want to make that model just do the exact thing with artificial neurons. You can do that. There's always going to be some caveats that make it not an exact replica. Or you can just talk about general statistics, like on average, this cell type is connected to that cell type with some distribution, and I'm going to replicate that in my model. So an example of talking about kind of this exact replica type, this is from a review article on this idea of building models based on connectomic data. And you can see here that there are, the things that you extract from the EM data to get to a point where you are building a model. You can get things like, certainly you can get kind of the binary connectivity matrix that you could put into your model. So just what cells are connected to what. You can also potentially have synaptic weight data or cell type data, excitatory inhibitory relationships. And so you can kind of pull that out. And as I showed with the artificial neural network, there is just really a direct way to put that in. You can put the weight values into an artificial neural network. An example of that being done is in this paper that uses the FLY visual system, the connectome from the FLY visual system to build basically a model that kind of is constrained by the FLY visual connectome, and then there's a few layers of learning to do a motion detection task. And then they note that when they initialize the connections with the true connectome, they can replicate some properties of the real neurons in doing that. And then, so I'll note, this and a lot of the papers that for now focus on using real connectome data are a bit more focused on using it to build a model and then compare it back to the brain. There are some people who try to use connectome data, obviously, to build better AI, but there are maybe reasons why that's not the most efficient route right now, and we can get into that. But certainly there is this mechanism for putting connectomic data into artificial neural networks. On the general statistics side, we can look at kind of things that people naturally pull out of the connectome when they have it. So for example, you can talk about motifs. So that's just kind of patterns of connectivity. If you look at 3 cells in your network, how often are they connected in these different ways? And you can look at a distribution of those motifs, how frequently they show up in the data. And basically people have identified kind of these histograms that show that, like the C. elegans is not a random network. If you just pulled a random network, it would have a different distribution of motifs than we actually find in the biology. And so you could build an artificial neural network that is at least initialized with the distribution of motifs that we see in the real data, and maybe hope that that helps it accomplish some task more efficiently than if you actually started with a random network. Another example of kind of general statistics is this paper that built something called MouseNet, which uses Allen Institute data, a lot of different data, some about connection probabilities, but also a little bit about the properties of the cells and things, to give kind of a broad overview of the mouse visual architecture and kind of how many cells from different areas should connect to other areas, and that kind of what resolution and that kind of thing. And so they're using not precise connectomic data, but broad statistics of the connectivity between different visual areas and different layers within visual areas to define an architecture and then train that architecture. So this is not defining the weights based on the connectome, it's just defining which cells are allowed to connect to which other cells based on broad connectivity data. So some of the difficulties with this direct approach of transferring the details of the brain to artificial neural networks is that basically to bring in the connectivity detail, you already have to have other detail present, basically. So in the generic artificial neural networks that people use in machine learning, there isn't different cell types. All of the cells are just this like basic ReLU, at least for most of these networks. And so the idea of saying, oh, this cell connects to that cell in the brain, well, now you need to define something as being those cells in the network, and you need to kind of give them special properties that make them identifiable as that cell, or at least it's smart to do that to actually have a kind of meaningful relationship between the data and the model. And so that requires adding this other detail to the model. And then the second issue is, as I said, these networks are trained to do tasks. And if you're starting with a weight matrix, the question is kind of how do you best incorporate that with the idea of training? Should you just let the network train in an unconstrained way and just reset the weights as it needs to? Should you try to constrain it to stay near the connectome? These are questions people have to fiddle with. So the second level of looking at the algorithms that the brain uses by studying connectomes, this has been, debated amongst neuroscientists for a while, this kind of idea of, oh, we've had the C. elegans connectome for so long, and has it actually taught us anything about their brain or how to predict their behavior or anything like that? There's this open question almost of like, what do we do with the connectome data that gives us an algorithmic level understanding of the brain? And so some modeling papers that have kind of weighed in on this, kind of what can you do with a connectome? What can you get out of it? There's this paper that uses graph theory to say, based on the motifs in the connectivity of a network, they can predict the attractor states of the activity of that network. So really kind of going directly from structure to function of some kind, like saying if neurons are connected this way, this is the activity they're going to produce. Now, of course, this can only be done in very simple model networks for now, but there is that kind of hope that you could look at a connectome and actually predict the activity that the network will produce. And in this case, attractors are computationally important for a lot of different things like memory or producing kind of central pattern generators and that kind of thing. So being able to look at structure and predict kind of the long-term behavior of the network is actually quite valuable as a way of understanding the algorithms that the network is implementing. Another paper looked at if you train these large artificial neural networks using a bunch of different learning methods, and then you want to say, oh, I don't know what I trained them on, kind of blind yourself to what you train the networks on and ask, can I look at the network and determine what it was trained with, what learning algorithm it was trained with? This was asking if you look at the weights of the network and how they trained over time, can you determine which learning algorithm was used? If you look at the activity of the network and how it changed over time, can you determine this? And then also if you look at kind of the transformation that individual layers in the network do, can you determine which learning algorithm was used? And they find that you can use the weights, so basically the connectome and how it changed over time. You can use that to get pretty good performance on determining which learning algorithm was used. This is just comparing two different learning algorithms. So chance performance here is 50%. So you can get up to 89% if you have access to all of the weights and there's no noise in your data. But you can see here that as you add noise to the data and as you are only able to sample a subset of the weights, that performance really falls off a lot to around chance levels if you're just looking at the weights. Whereas if you look at the activity, it actually is more robust to noise and subsampling. So in this case, they're claiming that actually here the connectome isn't as helpful in reverse engineering the learning algorithm that was used. Actually, activity is more helpful. And of course, activity is a lot easier to get, especially over time. So in this case, there's a kind of a conclusion that the weights aren't the best metric, especially if you can't get them perfect and you can't get them at multiple time points, get the full distribution of the weights at multiple time points. So that's kind of, modeling papers that have tried to look at this kind of thing, come up both ways. In very simple settings, the connections can tell you something about what the network is doing, but in more complex settings, the connectivity might be harder to analyze and actually pull out this kind of algorithmic level description from. So basically some of the difficulties in doing this identification, I didn't talk specifically about Eve Marder's work, but her work is really relevant here because she's used the connectome of a part of the lobster gut, and a lot of different modeling techniques to speak to this relationship between the connectivity structure and what the circuit is actually doing. And one of the relevant things she finds is that there is a many-to-one mapping between the connectome and the at least the activity that the network produces. And so that means that you can have a lot of different connectivity structures that in the end are producing the same activity. And so that complicates, this idea of finding this relationship. Also, what her work shows is actually there's a many-to-one mapping the other way as well, because you can have a single connectomic structure, a single circuit architecture, but based on the presence of neuromodulators, it could actually produce very different activity. And so that, again, just really puts a complication in trying to look at the connectivity to get the algorithm. And really, ultimately, if you want to get a sense of what the brain is doing by looking at the connectome, you just have to combine it with a bunch of other data and context, and basically you're just doing neuroscience then. So there's just not a shortcut from the connectome to an algorithm to AI. You basically just have to do all of neuroscience and then pull, insights from neuroscience to use in AI. So the connectome can speak to this end of the spectrum of relating AI to neuroscience, but getting those algorithms out is really difficult, and there are a lot of caveats in trying to just match the structure directly without adding a bunch of other detail that we would need to have to really make that structure have meaning in the context of an artificial neural network. Okay, thanks.
So thank you very much indeed, Grace, for that really brilliant synthesis. I'm looking forward to some of the discussion points afterwards that I think come out of that. So we're now going to finish with Tony Zador. So he's a professor at Cold Spring Harbor. I think Tony's had an unusual history, at least to me, in terms of his involvement in connectomics. I think he really started out as a systems neuroscientist and physiologist, and he has descended perhaps to molecular biology and connectomics, which is perhaps the opposite of many people's path. But Tony, I'm really looking forward to your talk, the third of these three. Over to you.
Can you hear me?
Yep.
Okay. So yes, in fact, before I became a systems physiologist, I was actually a computational neuroscientist and neural network researcher. So I've really come full circle. I just wanted to thank you guys for this fantastic opportunity and this great session. This slide basically summarizes a lot of what my lab has been doing for the last 10 plus years. We've been trying to figure out a way to grind up brains and convert them into neural circuits. And I'll say a little bit about that at the end. But a lot of my talk is going to be focused on a sort of zoom out to 30,000 feet, or I guess given the international audience, 10,000 meters point of view, and ask why I think connectomes are important for AI. And I think the previous two talks have been great at sort of setting the stage for my more speculative talk. So actually one of the main things that drove me to become a neuroscientist was I watched a lot of science fiction as a kid. And these were some of my science fiction influences. This is actually a cultural test that sometimes I give, but these are, the question is, why can't we build these human level AI systems? Why aren't we there yet? And how do we get there? And when I started graduate school, the dominant paradigm was what's now referred to as good old fashioned AI. And the idea is that you start with those things, the most interesting problems in AI are those problems that are uniquely human, like high level cognition, playing chess, language. Then you move on to the somewhat so-called simpler problems like reasoning, which a handful of animals can seem to do, at least to some extent. And for the most part, you ignore the problems, the kinds of tasks that the vast majority of animals and that could be called sort of human or sapiocentric AI. So to push back on this, even 30 years ago, AI pioneer Hans Moravec once wrote, encoded in the large, highly evolved sensory and motor portions of the human brain is 1 billion years of experience about the nature of the world and how to survive in it. the deliberate process we call reasoning is, I believe, the thinnest veneer of human thought, effective only because it is supported by this much older and much more powerful, though usually unconscious, sensory motor knowledge. We're all prodigious Olympians in perceptual and motor areas, so good that we make the difficult look easy. Abstract thought, though, is a new trick, perhaps less than 100,000 years old. We have not yet mastered it. It is not all that intrinsically difficult. It just seems so when we do it. And so in some sense, what he's saying is that what animals do is hard, cognition and reasoning are the easy part. And this has led to, I would say, this is leading to a version of the Copernican revolution, where, as you remember, Copernicus took our focus away from ourselves, putting the Earth, and really the important thing about the Earth was that we are on it, and toward the sun. And in the same sense, I think AI is now at the verge of a Moraveckian revolution, where instead of focusing on humans as the most interesting topic for artificial intelligence, we're starting with the things that animals do, zoocentric AI, interacting with the world, which turns out to be extremely difficult for AI. And then moving on to the somewhat easier, once we have that foundation, we can move on to the somewhat easier types of problems like planning and reasoning. And then once we get these things as a solid foundation, the theory is that the things that are uniquely human, and there are some things that are uniquely human, such as crying out of emotion, we can move on to those, but with a solid foundation, we'll do it the right way. So our goal is to achieve mouse level AI. I think that's an achievable goal. And that mouse level AI would provide a solid foundation for moving on to human level AI. So the outline of the remaining part of my talk is, first of all, to describe how modern AI relies mostly on learning, whereas animals rely on innate structure, to talk about the fact that the innate structure arises from the genome, which specifies a wiring diagram, and that the wiring diagram is compressed through a genomic bottleneck, and then to talk a little bit about some technologies that my lab has been developing, MapSeq and BarSeq, which enable higher throughput anatomy based on DNA sequencing by converting the problem of connectomics into a problem of DNA sequencing. So as Grace already pointed out, AI really relies on learning. The typical paradigm in so-called supervised learning is to take a large data set of labeled pictures, maybe millions or tens of millions of pictures, and then use an algorithm called backpropagation to extract, to squeeze out the structure of that and put it into a synaptic weight matrix. And in these algorithms, typically the networks start tabula rasa. So we need huge amounts of labeled examples to learn all the parameters. But of course, that's not how humans work. Labeling objects every second for a year would be of only 3 times 10 to the 7 training examples. And for those of us who've had kids, it does seem like we're labeling objects every second for 3 years, but that's actually an overestimate. So this is clearly not how humans or any other animals do it. And this has been well recognized in the field. In fact, Jan LeCun famously said that if intelligence is the cake, the bulk of the cake is unsupervised learning, the icing on the cake is supervised learning, and the cherry on the cake is reinforcement learning. So he's saying, yeah, it's still all learning, just not the kind of learning that traditionally is used in artificial neural networks, namely supervised learning. These other two are not as data hungry. But it's clear that even unsupervised learning isn't enough. Squirrels learn to jump tree to tree pretty effectively in a couple million seconds of life. A colt can stand essentially at birth, and spiders can hunt at birth. And then there are some things that are complex behaviors that aren't learned at all, like termites build termite mounds, birds build characteristic nests, beavers build dams without instruction. And in fact, if you're interested in hearing or reading about or watching beavers build dams, I recommend the YouTube page Justin Beaver, the Rescue Beaver, where a woman adopted a young beaver who had lost its parents, and the beaver showed characteristically beaver-like behaviors in her home, as you can see from the damage the beaver did to the walls and his attempts to build a dam inside the bathtub. In fact, Hopi Hoekstra at Harvard studies what you might call this a similarly innate behavior. Burrowing in Paramiscus, there are two species that are very closely related, except one builds, the old field mice build a long escape tunnel, whereas the deer mice build a very short one. And it turns out through cross-fostering experiments that the kind of burrow they build is genetic, not learned. So you can take the pups of one and have them reared by the other, and they'll build the kind of tunnel from their biological parents. So it's very clear from an evolutionary perspective why we want to build in as much innate behavior as possible, which is that, you know, the moment a pup or any kind of a youngster is born, a clock is ticking, and the faster that creature can reach maturity and reproduce, the better off it will be, and the more likely it will be to survive to the point of reproduction. So the idea is that it's quite clear that you want to build in as much innate behavior as possible, not too much, because you want to retain room for flexibility to deal with the world. Evolution will typically maximize fitness at birth by incorporating innate structure. So basically, I'm pushing back at the picture that many people in the AI community have, which is the brain basically comes prepackaged. It doesn't arise mainly through learning. It comes prepackaged, and as an American, I will focus on this form of prepackaged cake mix. Okay, summary so far: AI relies on learning, and natural intelligence relies on innate strategies. So where does this innate structure come from? It comes from the genome. So basically, genes specify wiring diagrams. The innate behaviors are encoded somehow indirectly in the genome. So the genes specify the wiring diagram. So how many parameters does it take to specify a simple brain? We've already talked a little bit about C. elegans, which has 302 neurons and about 7,000 synapses. And in that case, there's really no challenge. The genome of C. elegans is about 100 million bits, and it's easy to specify the connection diagram of a 7,000 synapses arbitrarily in 100 million bits. No real challenge there. Every connection could in principle be explicitly specified in the genome. But for the human brain, it's a lot more complicated. Naive calculation says that, well, if we have of order 10 to the 11 neurons, here, I'll just go to the more complex, or the more sophisticated calculation basically says that we have about 10 to the 15 bits per brain if you exploit the sparsity of human connectivity. But that's a far cry from the number of bits that we have available in the genome. Even if we use every base pair to try to hand wire a connectome, we'd still be 6 orders of magnitude off. So it's obvious that the human genome couldn't possibly specify the detailed connections of a human brain. So this is well known. It's not really a mystery, but the fact is there's a genomic bottleneck. Whatever is specified about the human brain needs to pass through this genomic bottleneck, this compression, if you like, of the wiring diagram. And the reason, the way that the resolution of this mystery, or this paradox, is that you don't specify every connection in the brain synapse by synapse. Rather, there are rules, and these rules can be far more compact than the hand-wired specification. So in a C. elegans, you could, in principle, specify every connection, connection by connection. But for example, let's say that you had even a very large grid. It would take very few parameters to specify the simple rule that each neuron should be connected to its 4 neighbors. Now, that's not a very interesting circuit, but with more complex rules, we can apparently specify circuits that are interesting enough that they enable humans to do what we do and dogs to do what they do, like chew through my internet cable this morning. And so the actual Kolmogorov complexity of the wiring diagram of the brain at birth is much less than the apparent complexity. And biologists recognize this fact, and that's why we study neural development. So the summary of part 2 is that AI relies on learning from examples. natural intelligence relies on a genomic compression of this W. And if I had more time to talk about this, I would go on to describe how the genomic compression actually is an algorithmic advantage because it forces evolution to squeeze out what's important, what it's extracting from the environment, and thereby provide better generalization. In other words, to enable the animal to interact with its environment in a more effective way than if it merely memorized everything that had ever happened to it. Okay, and there are 5 minutes. I'm going to talk a little bit about some technologies that my lab has been developing to enable a lower resolution and higher throughput method of assessing brain structure and wiring that's, I would say, complementary to the EM connectomic approaches that you've been hearing about, that you heard about earlier. And really, the idea of this approach is we'd like to be able to, in a single brain, in a repeated way, look at connectivity, and that's through a technique we call BARSI. Look at activity, let's say through a method like two-photon microscopy, and then also look at gene expression within the very same neurons from which we've learned about connectivity and function. So what we'd really like to do, I'm not going to tell you about all this today, but what we'd really like to do is to be able to take an animal performing a task. So my lab studies auditory processing. So an animal that's performing an auditory decision task, to record a lot of the neurons in that. And then for each of the neurons whose activity we've recorded, to figure out its connectivity and gene expression to understand how it's embedded in the circuitry. So as we've already discussed, the challenge of circuitry is that the axons are all tangled together, as shown in this picture. And so it's very hard to trace any individual one unless you use a method like electron microscopy. There is another approach that was introduced about 15 years ago now by Jean Livet, Josh Sainz, and Jeff Lichtman. And there the idea is that we can trace axons in principle more easily if we could give them different colors. And they came up with some very clever genetic tricks to enable theoretical diversity of 200 different colors to be expressed in this bunch of neurons. The challenge of this technique, though, is that although in principle there are 200 colors, it's very hard to resolve that many colors. In fact, it's often hard to resolve more than 4 or 5 colors. So these 3 colors purple are likely different, but they appear to be the same. So our solution inspired to that is to replace colors with numbers. And that's the basic idea behind MapSeq. We have a unique DNA barcode inside of each neuron. And of course, the theoretical diversity there approaches 0 as the length of the barcodes become longer. And so very briefly, the technology developed by Ian Pycon, Eustace Kepschel, and several others in the lab was to make a library of barcoded viruses, each one expressing its own individual virus, and inject it into a part of the brain along with a carrier protein that drags the barcode out to the synapses. And that was really one of the more challenging parts of this, is to drag the RNA barcodes out to the axons and to the synapses where we need them. And so I'm just going to touch on a handful of the things that we can do with that. So if we really scale that up, Mengwen Huang showed that we can actually measure the projections of 100,000 or more neurons per brain using this technique. And here are the projections of 2 of these neurons out of that 100,000 that we got in this brain. The limitations of MapSeq, though, very briefly, are crude spatial resolution. We can't see much about the individual neurons, and we can't relate it back. So we can't relate it back to gene expression, and we can't relate it to activity either. And so for that, we developed a next generation version of MapSeq called BARSeq, which involves in situ sequencing. And very briefly, the idea is to repeatedly image a neuron that's expressing this barcode, where for each round of imaging, just like in a sequencing machine, we can read out the barcode. And so here is a successive series of images of some neurons that have been barcoded. And you can see that the same neuron gets a different color for each round depending on the barcode in it. This can be used for axonal tracing in vivo, it can be combined with gene expression. And so in summary, AI relies mainly on learning, whereas animals rely on innate structure. Innate structure arises from the genome, which specifies the wiring diagram. The wiring diagram is compressed through a genomic bottleneck, and MAP-seq and BAR-seq represent a path to high throughput anatomy based on DNA sequencing. And I'm going to skip that and just do a plug for our NeurAI Scholars Program at Cold Spring Harbor, where we are looking for people with advanced training in artificial intelligence and embed them in a neuroscience group so that we can take insights from neuroscience and apply them to AI. And with that, I'll close with some words of wisdom that I always turn to when thinking about the brain. If the brain were so simple we could understand it, we would be so simple we couldn't. And I'll also thank my collaborators and my funders. And I went only 1 minute over, so thank you for your attention.
Thank you very much indeed, Tony, for that really exciting and wide-ranging talk. So our instructions now are to have a combination of panel discussion and also to respond to questions which have been coming in through the Q&A. We'll keep monitoring the Q&A to pick out from questions from there. I thought I would start, if possible, by focusing on sort of a few topics suggested by the questions at the level of synapses and maybe at the level of networks, and then maybe we could start to talk about their relationship to AI. And I just also want to say a big thank you to the 3 speakers today with 3 really nicely packaged short talks that set this up. So thank you very much indeed, and for staying on good time. So to start us off, maybe I could ask on behalf of the Q&A a couple of questions that relate to synapses. And perhaps we should start with you, Moritz, for these. I don't know if you want to unmute. So the first one is, can you identify synapses as excitatory or inhibitory from EM images? And then the second question, moving on from that, is, are there visual differences between synapses that are capable of learning and those that are fixed?
So E and I, at least in the mammalian brain, is luckily reasonably detectable, sometimes at the single synapse level, but we think, or we rely heavily, especially on axonal classification. So using the fact that if you have an entire axon with tens of synapses, ideally, or even much more, many more, then you can use the combined evidence from these many synapses to classify the axon and then propagate that back into each of these synapses. So I think E and I in the mammalian brain is reasonably doable. It's more difficult in other species where, of course, first of all, more transmitters exist, and then also based on the postsynaptic receptors, things can be different. So these are things that are much harder to identify, although colleagues are also working on that, obviously, in the field to try to even pull that out of images. In terms of learned versus not learned, I mean, this is something we're trying to understand better. There has been the suggestion that, again, the mammalian cortex organelles like the spine apparatus and others might be indicators of learning. We haven't so far seen any simple relationship, but this is clearly something that people are very interested in. So far we have to rely on these analyses that we proposed.
So maybe I could then just open that little theme out for a little bit more discussion. So Moritz, I think you very nicely, sort of compared the idea of having a part of the connectome which might be fixed, which sort of encapsulates evolutionarily learned information, again, in the way that Tony was echoing later, and Grace also during her talk. So if you look at the sort of networks that come out of EM connectomics and maybe in due course molecular connectomics, do you think there are going to be parts of the network that are obvious, that you can tell are the fixed parts, and parts that you can tell are the learned parts? And so then I guess the question for Grace, for example, would be, how useful to you in a modeling sense do you think it would be to know which parts are learned and which parts are fixed? So it's kind of a two-part discussion where it's, what are our chances of extracting that at sort of network level? And Grace, do you think this could be useful information?
Yeah, just briefly, I mean, we think that as long as learning models make clear predictions about weight distributions, then tricks like the pair synapse approach can be used to extract at least possibly learned configurations. We have to be careful, of course, but you could think about that more in a more complex way, that essentially you do entire models of the evolution of synaptic weight in the network, and then ask whether any snapshot connectome provides evidence towards one or the other. We had this a little bit pessimistic study cited by Grace about that. I think it's the situation is much more benign the more you know already about how the network should be configured, and by exploiting things like multiple synapses per connection. So yes, I mean, we think we exemplified as a first attempt how to do that in a network map, the possibly learned fraction. But this, of course, I hope can be extended, especially when the networks become more complete and larger, and then you can really disentangle that even more from the, genetically imposed connectivity.
Yeah, I think that kind of data could definitely be interesting. As I said, it's unclear at the moment how to best combine connectomic data with learning that has to happen in these artificial neural networks if you want them to actually get something interesting done. Basically, you can't just throw in the connectome and then have it work. It doesn't work out of the box like that. So you need to do learning on top of it. And so if you had a sense of which synapses in the model should be most the candidates for learning, and others should be more fixed, people would definitely try that out at least. And so there's also, we kind of need to speak to the two different goals of computational neuroscience versus AI. So as a computational neuroscientist, that data is super helpful. I can fiddle around with that and try to make a model that captures other data and all of that. As a person building an AI model, then the question is just what works. And for that, it's very much an empirical science. You could try to constrain parts of the connectome you think are innate and let the other parts learn. But if you get better performance by letting the whole thing learn, that's what you're going to do. And so there are just, different goals here, and reasons why the most biologically detailed, accurate model might not be the best AI model right now. Because as I said, there's kind of all these other details and constraints that we don't have in the AI models. And so you can't just throw in a detail and assume it'll make things better, because a lot of these things kind of work in unison with other biological details.
If I could just add a couple things to that. I think that it may not be the case that it's so easy to say that these synapses are built in and then these are plastic. I think that instead, at least in a lot of systems, the way it works is that development specifies a probability distribution over connectivities, and that the way you're going to be able to get that probability distribution is to look at a relatively large number of samples, because that's the only way you can sample through a distribution and infer, what the, if you like, covariance matrix, or what the correlations are, connections. And I think the other thing that I would say is that in a lot of biological networks, connectivity remains far, far sparser and more structured than in artificial networks. And that sparsity is really crucial. And there's actually a NeuroAI fellow here who has taken seriously the wiring of C. elegans as the basis for a reinforcement learning task called the swimmer, and has been able, this is Nikhil Bhattasali, who's, you can look up his work, and he's shown that by taking that seriously, you can accelerate the learning of this classic reinforcement learning task, the swimmer, by a considerable amount. So I think there are lessons to be learned from a lot of these connectomes, even today, potentially, for some of these tasks.
So thanks a lot for those initial comments. Maybe I could just follow one direction implied by a couple of questions there, which were asking about network properties. So I don't know if any of you would like to summarize a little bit, for people, what is sparse when you're talking about sparse in real neural networks. So I guess, in principle, if you have a set of n neurons, you can potentially have n squared sets of connections to think about between them. So what's, what is the subset of all possible questions that are actually realized or possible connections, sorry, that are actually realized in real brains? That's probably, a question firstly for Moritz or Tony.
Well, I'll just say that, I mean, naively, if you have 10 to the 11 neurons, right, then you have, in principle, 10 to the 22 possible connections. But in fact, each neuron typically makes only about 1,000 connections. So that's already a dramatic decrease in the number of possible configurations by, I think 7, 8 orders of magnitude. And that's the most naive consideration. There are further constraints that most neurons connect locally. So if you start building those things in, the actual possible circuits that can be wired up in a real brain are, a tiny fraction. The level of sparsity is really dramatic. And I although there are artificial neural network approaches to achieving sparsity, I think none of them operate in anything like sparse regime, or for that matter, that degree of structure.
Moritz, would you like? Oh yeah, go ahead, please, Grace. Sorry,
yeah, I can speak a little bit to kind of the general architectures of the artificial neural networks, just to give a comparison. So, you can have deep neural networks that have thousands to millions of units, but they're not all-to-all connected either. A common separation is to have these different layers, like I kind of showed in the example architecture, where neurons in one layer only project to neurons in the next layer. But there you'll have all-to-all connectivity, or at least you'll have you'll initialize it that way, and the network can learn to kind of disregard certain connections and set them close to zero. But you have the possibility for every neuron in that layer to connect to the next layer. And if you think of that as kind of 2 different brain areas, we don't expect all neurons in one brain area to connect to another brain area. So that is not as sparse as you see biologically. And there's also different kind of structures that impose other forms of local connectivity. So convolutional neural networks are used to study vision a lot. And in those, a neuron at one layer doesn't get input from the neurons, from all the neurons at the layer before. It only gets input from a select region of those neurons that represent a select region of the visual space. And so this is replicating receptive fields in real neurons. So there are constraints that people put in, some inspired by biology, and also just because it's for the same reason that it would be tough for the brain to have connectivity everywhere. It's also difficult for artificial neural networks to do that, especially when you're training those connections. The number of connections is the number of parameters. You have to train more parameters, you usually need more data. There's a lot of reasons why people in ML want to reduce the number of connections in some kind of principled way beforehand as much as they can.
Yeah, maybe briefly 2 points. So one is in terms of circuit configuration space, I would like to re-emphasize from my initial slides. It's a huge difference if you think about cell type based connectivity or individual connectivity. So if you only quote unquote assume cell type based connectivity, then the configuration space is much, much smaller, of course, than that you can add if each neuron can essentially decide if you want on its own who to connect to or not. Now, obviously, there will be a mixture between the two. There will be biases to connect in one way or other that might be cell type based, and then particular implementations that are individual and so on and so on. But this, I think, is very important to realize. And I think that the cell type based connectivity is something that there are many ways to get at. The individual connectivity is very hard to get at, and EM is one way of doing that. And then it depends on species where one is more important than the other, I think. So it clearly is, I mean, in C elegans, you could say cell types equals single cells. Almost. Yeah? So these 302 neurons have an identity, and with a few maybe symmetric exceptions, it's essentially each one matters, right? as much as it possibly can, with the advantage of being able to overspecify each one, each synapse even, but also with a indication that there may be very little individual neuron level learning in that sense, right? And then, of course, the other end of the spectrum, it could be also relating to Tony's slide, that maybe there is actually a phase in evolution where animals have most pre-learned, so genetic knowledge in the circuit. And then maybe humans have reduced that again because we want to be flexible, right? So this trade-off between existing knowledge and pre-specification and ability to adapt is, of course, also there. So that is, I think, something that we really need to figure out. And that matters a lot when you talk about the configuration space. And just very briefly about sparsity, actually deep feed-forward only networks that we're using for AI vision currently all the time are actually super sparse, right? Compared to, because they don't have intra-layer and they only have cross-layer and not jumping layers, at least in the most implementations. That's super sparse compared to the full connectivity. Our brains are somewhere in between, and especially they are highly recurrent. And this combination of recurrency and sparsity, I think, is really something very special that we haven't understood.
I should say also, because I think it relates to a question that came up, you can have recurrent artificial neural networks. I just want to make that clear, because I only showed a feed-forward structure, but people use recurrent structures in artificial neural networks as well. You can basically define any architecture you want, and that's part of the problem. So might be good to constrain those with biological data.
So maybe just to follow up a little bit in more in this area. So there were questions about whether, there's sort of something special and defining about the network architecture of a connectome based on what we know so far. Presumably right now it would be pretty easy to distinguish an artificial network, read off from a particular AI solution versus a brain network. Could you say any more about, the properties that are distinctive other than sparseness? Recurrence sounds like it's, is there a lot of difference in the way in which recurrence is currently implemented in artificial networks?
I mean, I would say one of the relevant things to remember when doing this comparison is that you don't usually have different cell types in artificial neural networks. So it's hard to actually say, how would you compare this? If you were not labeling cell types in the real brain, you would probably still see these kind of emergent trends that are the result of different cell types connecting with different probabilities, but you don't have that kind of modularity or those groups to think about in the artificial neural network. So that's just a big difference that right off the bat is going to make the connectomes look different.
So there are, many neuroscientists think about many different cell types, and those might have different rules of connectivity, but they also might have, important differential rules relating to dynamics as well. So Grace, in your really nice synthesis, you mentioned, work from Yves Marder, which I think is pretty much classic at this point. Showing how potentially the same set of connectivity can generate different dynamics. And one of the things that she's really emphasized there is how neuromodulation, so sort of quite broadband signals, can change circuit configurations dynamically. So I was just wondering, in AI, are people trying to use, neurons, nodes that actually might have distinctive dynamic properties like that, or broader impacts on circuit configuration?
Right.
Sorry?
Go ahead.
Yeah. I was just going to say that so when people talk about kind of different unit types in artificial neural networks, there are different unit types, but it's not the same as different cell types in the brain. So when people in AI use recurrent neural networks, they usually use like a gated unit, which has some kind of internal machinery that helps hold on to memory for longer. So there is a slight notion of, different cells have different dynamics in that sense, but it's not, it's so relatable to different cell types in the brain, and you don't usually see like one neuron that's kind of a mix of all different types of circuits. It's kind of like you either have a regular feed-forward network or you're using a recurrent network, and it has these different types of units. So there is already a concept of building in different dynamics, but yeah, not so easily relatable to different dynamics in biological cells.
Tony, sorry, I think you had something to add as well.
Yeah, I was just going to add that, one of the roles of neuromodulation can be to provide a more global signal to the circuit. And that's something that certainly is often used. There are global parameters, global signals that get spread to an artificial circuit. But I think that for the most part, most artificial neural circuits don't really exploit dynamics to the extent that natural circuits do. And it sort of becomes hard to take some of the lessons that we might want from connectomics and bring them to artificial circuits, because the fundamental dynamics of most, especially non-spiking artificial circuits are so different. In fact, there's a workshop right now on spiking neural network dynamics. And for the most part, this is sort of a niche field in artificial neural networks. People don't really know how to think about spiking, or it's been pretty challenging to demonstrate that it's useful for anything. And so it's really kind of a corner. And yet, I think a lot of what we see in connectivity is driven by the need to achieve the right circuit dynamics. So there's sort of this gulf here that perhaps one day we can cross.
Yeah, and that's a great example of just this idea that like these details relate to each other. You might have neurotransmitters that work a certain way because it's a spiking network, and you shouldn't assume that putting that into a rate-based model is going to give you the same benefit. Like you really have to think about all these interactions.
So there are quite a few questions that relate to this sort of, innate versus learned aspects of connectomes and even behavior. I guess maybe just one last sort of discussion point there. So Moritz introduced the idea of the cell type connectome being much smaller than the rest of the connectome. I'm curious for you, Tony. Do you feel that sort of solves the genomic bottleneck, just at least in terms of how much information might actually need to be encoded? And maybe just to put both of you on the spot, do you have any notion of how many cell types, by whatever definition, a mouse or a human ought to have in its brain?
Yeah, so I think cell types are clearly part of the solution to how we are able to squeeze enough circuit information into a limited genome, right? As Moritz pointed out, if your rules are of the form connect, cell type A to cell type B, that already sort of sets up for far more efficient rules. I'm not sure that that's all of it. In terms of how many cell types there are, it's clear that there are some at the top level very discrete types. Neurons are different from non-neurons. Excitatory neurons are different from inhibitory neurons. In the cortex, you have three main types: IT, PT, CT. but we can keep subdividing. And at some point, although cells are continue to be different, it's not clear that discrete cell types are really the best way to think about it. Another way to think about it might be that you have combinatorial patterns that enable you to wire the system together better. And these combinatorial patterns are exploited through development. And what we see in the adult, when we look at transcriptomic cell types, are sort of the echoes of the developmental program. So at some point, I think maybe the search for n discrete types might be misguided. And I say that because in my day job, we've been searching for n discrete types based on combining gene expression and connectivity. And although there's a lot of diversity and there's a lot of structure in the diversity, the particular data structure which leads to discrete types is probably not the best way to capture that diversity. But that said, it's probably hundreds.
Yeah, hundreds. Okay, Moritz, do you want to?
I think it's a very relevant question and debate, and I think I agree that the notion that it will be very clear is not necessarily to be expected beyond simple types, especially because if you think about it, generatively, there will be, of course, some genetic programs that drive specializations that also show up at morphology, ethos, obviously gene expression and circuit levels. But then there might be homogeneous, if you want, genetic populations that by virtue of the data that comes in, so to say, are driven into separate connectivity regimes, that might separate their activity by not connecting to each other and so on to represent kinds of stimuli that are very similar and should be separated or something like that. So essentially there could be connectivity patterns that are really learned, individually learned, enabled by the genetic program, but individually learned. And then, of course, this is now a little bit extreme, but there could be in between stages. And that could be one of the reasons for gradients of connectivity, that you do start with a genetically pre-specified possibility of wiring. But then, of course, sensory information, at least in mammals and animals like that, shapes these and uses these. And then it becomes at some point difficult to differentiate, is this now a different cell type or just another version of something that has been set up before? And then it becomes, I think, useless to talk about cell types or not. It becomes interesting to then talk about the actual connectivity structure.
I think that's a point that also came up in some of the questioning in terms of, is there really a gradation between these notions of, learned versus innately specified types? And I guess it's likely there is. So we're just coming up to the half hour now. So I just want to finish by picking out maybe one or two questions that are sort of distinct from the topics we've discussed so far. So one of them is, what do you think the road is to applying this kind of connectomic research to more broad neurological or even psychiatric diseases, Alzheimer's, schizophrenia, bipolar disorder? Do you see, there's a lot of work maybe to do before that's possible, but do you think it's going to be useful anytime soon?
Yeah, I mean, I can briefly say I absolutely think so. And I think it's sooner than later because we do have many models. We can screen connectomically at the M level by now, and we are also doing that. And it's also important to look at human tissue, also possibly diseased human tissue, and start learning the variability and start learning about what can change and cannot change. And I think this is absolutely pressing to apply these techniques there.
Yeah, I'll just add that I'm a full disclosure, a co-founder of a company that Cahal Neuroscience that exploits some of the technologies that my lab developed and other labs developed in order to gain insights into neurodegeneration from connectomics. So I think the answer is yes, and some VCs agreed.
Okay. I think I'm just looking at the time and thinking maybe this would be a good moment to bring the discussion to a close. Thank you so much, everyone, for the questions that you put into the Q&A. We've tried to synthesize quite a lot of those into the discussion. So once again, a big thank you to our three panelists for making some talks that are both exciting and cover sort of frontier research in neuroscience, and including in connectomics, but also making them accessible for this broad life sciences across the globe audience. So
thanks for being a great host there, Greg.
Not at all. Thank you for doing all the hard work, the three of you. So we're now going to move on to a session for Meet the Panelists. And maybe Sarada, are you ready just to do the transition there?
Of course. Thank you, Greg. And thanks to all the panelists for a truly fascinating discussion. I mean, we could certainly go on longer, but in the interest of time, we would be closing. But before that, on behalf of the Life Science Across the Globe organizers at Janelia and all our partnering institutes, we would like to thank the speakers, Moritz, Grace, and Tony, for fantastic talks, and to Greg for beautifully bringing the entire session together. We really enjoyed the session. I'm sure the audience did as well. Audience, thank you for joining us today, and also for engaging in the session with some wonderful questions, which I guess added to the discussion as well. Please join us next month for another Life Science Across the Globe session hosted by the Cold Spring Harbor Laboratories. We would be addressing yet another relevant topic on sustainability.